In this lecture, we are going to cover the pharmacology of opioids, so let's get right into it. Opioids, sometimes called narcotics, are a group of drugs that act on the central nervous system to produce morphine-like effects such as pain relief and euphoria. Now, in order to gain better understanding of their mechanism of action, first we need to talk about the transmission of pain. So, pain begins at the nociceptors, which are simply the branching ends of sensory neurons found within the peripheral nervous system. These high-threshold primary sensory neurons respond to damage to the body by transmitting the painful stimulus to the second-order neurons in the dorsal horn of the spinal cord. From there, the signal is carried through the spinothalamic tract to the thalamus, and then to the somatosensory cortex where pain is perceived. Now, on a microscopic level, the pain signal takes the form of a series of action potentials that fire repeatedly depending on the intensity of pain. To enhance movement across the synaptic cleft, transmitter chemicals are released from the presynaptic neurons, including glutamate, substance P, and calcitonin gene-related peptide, CGRP for short. Glutamate is one of the most important neurotransmitters for pain and can activate both NMDA and AMPA receptors, which permit influx of positively charged calcium and sodium ions respectively. As you may recall, the flow of positively charged ions into the neuron makes the neuron more likely to fire. In this way, glutamate excites the second-order neurons in the dorsal horn, which leads to propagation of a sharp localized pain signal. Substance P, on the other hand, binds to the neurokinin 1 receptor, NK1 for short, which leads to intracellular signaling that involves activation of arachidonic acid pathways, nitric oxide synthesis, and activation of NMDA receptors. NMDA receptors are activated when substance P attaches to NK1 receptors and then gets incorporated into the cell, activating protein kinase C. This action removes the magnesium that under normal conditions is blocking NMDA receptor. This in turn allows glutamate to attach to the NMDA receptor and thus permit the inflow of calcium ions, ultimately causing the pain signal to increase and fire more frequently. Lastly, the released CGRP binds to its receptor on second-order neurons, leading to changes in receptor expression and function, and thereby altered neuronal activity. This in turn contributes to the so-called central sensitization that is characterized by a lower threshold for evoking action potentials. Now, fortunately for us, our bodies can cope with certain amount of pain by releasing so-called endogenous opioids. There are three major families of endogenous opioids: the enkephalins, dynorphins, and endorphins. Endogenous opioids exert their effects by binding to opioid receptors, which are abundantly present in the central and peripheral nervous systems. There are three major types of opioid receptors, that is mu, delta, and kappa. In general, all three receptors differ in their cellular distribution, their relative affinity for various opioid ligands, and their contribution to specific opioid effects. All opioid receptors are 7 transmembrane spanning proteins that couple to inhibitory G proteins, and they are all present in high concentrations in the dorsal horn of the spinal cord. Activation of these receptors by an agonist, such as the endogenous mu-opioid peptide endorphin, causes closing of the voltage-gated calcium channels on the presynaptic nerve terminals, which in turn decreases the release of neurotransmitters such as glutamate, substance P, and calcitonin gene-related peptide. In addition to that, activation of opioid receptors leads to opening of potassium channels, allowing efflux of potassium ions, which in turn results in hyperpolarization, rendering neurons less sensitive to excitatory inputs. Now, the majority of currently available opioid analgesics act primarily at the mu-opioid receptors, essentially mimicking the effects of endogenous opioid peptides. However, while naturally derived opioids can only reach a certain potency, the synthetically produced opioids are refined and processed to be much more powerful. The examples of synthetic opioid agonists are fentanyl, hydrocodone, Hydromorphone, Methadone, Meperidine, Oxycodone, and Oxymorphone. As a side note here, it's important to note that Methadone is not only a potent mu-receptor agonist, but also a potent antagonist of the NMDA receptor, as well as norepinephrine and serotonin reuptake inhibitor. These properties make Methadone useful for treatment of both nociceptive and neuropathic pain. Now, in addition to producing analgesia, activation of the opioid receptors in other parts of the body can bring about many side effects. For example, all opioids produce some degree of nausea, which is due to direct stimulation of the chemoreceptor trigger zone in the medulla. All opioid receptor agonists also produce a dose-dependent respiratory depression. Opioids primarily cause respiratory depression by reducing brainstem respiratory center responsiveness to carbon dioxide. They also depress